right, good afternoon, everyone. My name is Shanna-Lee Thomas, and I am the Chief Scientific Officer at the Discovery Bay Marine Lab, and I will be sharing the findings of a study to assess the reproductive activity of queen conch in captive and wild populations in the Discovery Bay Special Fishery Conservation Area in Jamaica. So as we might already know, queen conch reach sexual maturity at 3 to 4 years, and they are also aggregate spawners. So they are generally known to aggregate in higher densities around their breeding season, where they migrate to shallower waters to spawn, and this makes them vulnerable or susceptible to overexploitation. And if that wasn't enough, in areas where the adult populations have been removed, the sub-adults and larger juveniles are oftentimes removed before they ever get the chance to mate. Now, the National Fisheries Authority in 2018 confirmed that our local conch stocks were critically low. So it was sensationalized in the media that our conch fishery had crashed, but it was explained to me recently that what had happened was our stocks had approached a limit reference point as determined by the authority. And as a result, what they do is put measures in place to safeguard the remaining conch stocks. So they imposed a ban on the fishery in 2018, and that was for 2 years. And that was a closed season, essentially. However, outside of the 2-year closure, Jamaica's closed season is from August to February, and this is based on temperature-influenced annual spawning activity from other Caribbean countries. So no temperature-related reproductive behavior assessment and spawning season determination has ever been done for Jamaica. So this is where this project came in. So the aim of the project was to observe and document the temperature and period over which reproductive activity of Jamaica's queen conch takes place. So this project was carried out essentially in 2 different parts. So the first part was conducting abundance surveys in the Discovery Bay Special Fishery Conservation Area. So we established 6 sites. So 3 of those were on the eastern side of the bay and the other 3 on the western side. And what we did was try to get an idea of the distribution, the density, and the population structure of queen conch within Discovery Bay before starting the research. So this was done in 2020. And what this also allowed for was site reconnaissance, essentially. So we knew that we wanted to establish pens within the bay, but based on literature, we'd already know that conch prefer, during their breeding season, they prefer a mixture of coarse rubble, sand, and seagrass. So the abundance surveys allowed us to figure out or scout the area to determine where are the exact locations in which we place these pens. So we established a total of 4 pens. So 2 of them were located on the western side of the bay, while the other 2 were located on the eastern side. So we had 2 points. We had the jetty and the Fortlands Road point. So each pen measured approximately 50 meters squared, and they were built in order to allow for rotation. So once the conch had grazed one area, we would move them to the second or that backup pen in the same site. So we collected adult queen conch from within Discovery Bay. We sexed them, so we identified their genders by bringing them back to the lab. And we also collected shell morphometric data. So each conch was given a unique tag. So we used aluminum tree tags and assigned even number and pink yarn for the female conch and uneven numbers and a different color string or yarn for the male conch. And these were transferred shortly after to the in situ pens and stocked at a density of 2 to 3 male to female. We also deployed water temperature loggers, that's hobo pendant loggers, and these were configured to collect data at 2-hour intervals. The conch were monitored at a minimum of 2 times per week for reproductive activity, and we categorize this. So for each visit to the conch pen, we noted either mating as A, pairing as B, spawning C, and no reproductive activity as D. We also monitored conch in the wild for a similar time frame, just to compare their behavior to that of which is happening within the conch pens, the actual enclosures. So the results of the surveys that we conducted, the abundance surveys, indicated that the densities were low. They were at least half of the minimum density that's expected to bring about successful reproduction. Now, for the meat of the matter, we noted spawning or reproductive activity in both the captive and wild conch population. So for the wild conch, we noted spawning as early as April in the year, and that lasted until November of that same year. However, for the conch that were in captivity, we first noted spawning in August, and that continued right through October and November. And the photos that are currently on the screen indicate some of the evidence or proof that these conch were actually spawning or mating out at sea. The pictures indicate the spawning activity. So we had one conch spawning up to 5 times for the season. And you can also see where egg masses were laid in the pictures here. We also noted copulation in the wild, and that lasted, as I said, up to November. In terms of the temperatures, peak activity for the reproductive activity for the captive conch was recorded for August to October, and this coincided with the high seawater temperatures. So the temperatures gradually increased from May to September and decreased shortly after. However, the peak in the highest seawater temperatures were recorded between August to October, and that's when we saw most of the activity. So observation for the wild conch began as early as April and lasted until November. And we are aware that one study would not be sufficient to call for a revision of the close season. So in terms of future work, we would like to expand this study to other sanctuaries around Jamaica. And we've already started building capacity and sharing techniques. We did a workshop late last year, and we invited members from the Jamaica Fish Sanctuary Network, and they were educated on basic queen conch biology, and they are able to identify the different sexes and to identify egg mass. So even though we don't believe that all the sanctuaries would have the capacity to set up pens or enclosures to monitor conch, I think in order to determine the exact period in which spawning takes place, we could at a minimum set up data loggers and carry out monitoring of the wild population to pinpoint exactly when spawning starts and when it ends. I also think it's important for us to assess the conch population in other MPAs across Jamaica, because much is known about our conch stocks at Pedro Keys, but not much is known about what's happening onshore or in more localized areas. So it would be good. And last year we were able, through this project, to collect data on a few sanctuaries on the North Coast to find out where some of the aggregations are. So I believe if other sanctuaries are able to do this, then we'd be able to better safeguard our coastal conch stocks. The combined findings of this study will help to inform measures to manage conch stocks in SFCAs towards sustainable harvesting in nearby fishing grounds. And this year, we're hoping to also partner with the Florida Atlantic University to establish a mobile hatchery unit here at the Marine Lab, because a problem that we had with the first study is that the conch were poached from the enclosures frequently enough. However, with a hatchery, we can collect literally one egg mass and start repopulating the coastal conch populations. And that brings us to the end of the presentation. I'd like to thank the Jamaica Conservation Partners, the National Environment and Planning Agency, as well as the National Fisheries Authority for the support provided to complete this research. Thank you so much.
Yes, thank you very much, Ms. Thomas, for your presentation. And thank you for sharing these encouraging results. I heard you refer to, I guess, the media or perceptions about the conch fishery in the media, but there's a subtle thing that you mentioned there, and that is that the fishery was closed in response to a target reference point. That is not an easy thing to do as a country. You set the scientific limit and you stuck to it. So that has to be encouraged, and as a result, I think it sets an example for other countries in the region to follow. We are supposed to respond to the status of the fishery, and that is what took place. And I encourage you to stay the course. I see a hand by Mr. Kong. Please go ahead, Mr. Kong.
Thank you, Chair. I just want to congratulate Professor Weber, Dr. Trench, and the team to be looking at these critical areas of research that is so important for the management of one of the most important fishery in Jamaica. Just to say that, of course, you know that there the main fishery zone for queen conch in Jamaica is Pedro Bank, and there are differences within that ecosystem. I'm sure you're all aware of that. What came to mind is the issue of peak spawning. I hear you mention that you observe several spawning activity. Did you get a sense of any peak happening? I know it's a very small sample, but I would be interested if you saw any sense at all of any peak activity within a specific time. Thank you.
Yes, so prior to August, we weren't observing any reproductive behavior, no spawning, and the conch were in these enclosures for at least 3 months before we started seeing anything. So the peak in spawning activity that we noted was between August to September, and it started tapering thereafter. So even though we saw some activity in October, there was a decrease after September.
Okay, thank you for that answer, Mr. Kong. Please go ahead.
Just a quick follow-up. I just wanted to really add to your, I'd forgotten to just add to your opening sentiments. And just to say that Jamaica has always managed our conch fishery, ultra conservatively. And the other issue that a lot of people don't understand is that a fishery could have been affected during the time of the moratorium. But it's just that we were being ultra conservative at the time. I'm saying we because I was the director of fisheries at the time. And what would have been the reality there would be that the total allowable catch would have been minuscule and thus not viable at all economically at the time. But the population did not collapse as many people were trying to intimate. Thanks, Chair.